Very good afternoon to everybody. Now it's time for hearing a guest lecture from renowned forage breeder who has come as an external examiner for the PhD BIOS examination. I welcome Dr. Rahul Kapoor for having visiting our TNAU. And a brief introduction of the guest speaker is already it was informed to you by the Professor and Department of Forage Crops. So he's 15 years, more than 15 years, he has served in the forage breeding in PAU, Ludhiana, and he was deeply interested in understanding the genetic architecture of forage crops. And he is employing a lot of molecular tools for the improvement of forage crops. So with this experience, he will be delivering the lecture on forage crop improvement using biotechnological approaches. And he has developed 25 varieties in different forage crops at both at state and national level. And he has also contributed for the oat variety release, OL 1861. And he developed the first dual variety of oats, OL 1876-2, and in the country. And he reported the first report on the QTL mapping of beta glucan in oats. And he was also responsible for the first report for the QTL mapping for Helminthosporium leaf blight in oats. And he was the first time in the country he reported the somoclonal variants in Bajra Napier hybrid, and also first to report the Colchi diploidization event in Bajra Napier hybrid during 2017. So he has published more than 60 papers, and he has also taken the course on the fundamentals of genetics and plant breeding to BSc Agri, and also for the PhD students. And he has, as a major advisor, he guided 5 MSc and 2 PhD students, and he is also the advisory committee member of so many students. And he has also got several awards: recipient of University Merit Scholarship, recipient of Sardar Kartar Singh Dhillon Gold Medal for scoring highest OCPA during MSc in plant breeding genetics. And he has also contributed for getting the best AICRP Center of Forage Crops by ICAR. So he has received a lot of appreciation certificate. With this brief introduction, I invite Dr. Kapoor to deliver the lecture, and I'm happy to invite you. Thank you, sir.
Thank you, Chairman, sir. A very good morning to all present here, dear students. So we kept deliberately today's topic as the biotechnological interventions in forage crops. So you all know, you all are clearing biotechnology courses during your MSc PhD. So you are aware of various biotechnological interventions, various biotechnological techniques, tools available with the scientists nowadays. Forages are always considered to be orphan crops. Not many of you are ready to work with the forage group because these are the orphan crops. Everybody wants to work on major crops. In Punjab also, everybody is running for wheat, rice, and pulses. Here too, I think the same scene will be witnessed, or Dr. will be witnessing. So, but for the last few years, the scene has changed in many of the institutes, particularly in PAU also. The scene has changed. Even the toppers, they are asking to work with us in forage crops. So that's a kind of blessing to our forage groups that we are working in a sense that students wish to work with the forage crops. So it's overall very good for the forage farmers, dairy farmers. If the top students or the good students come and work and they publish good work or they generate a very good material, like today we saw a very good BN hybrid has been generated, which will go a long way in serving the dairy sector of your city or your state. So Co5, Co6 story, you know, it's a great success story. And we always give example to our students that just one hybrid, a university is selling 2 crore worth of rupees planting material. It's not a menial task. It's a daunting task. It's a challenging task. Single variety planting material is being sold worth of 2 crore rupees is a great achievement. So such things can be done. They are not far off. They can be done very easily if you are doing really good work. So this is the success story of your center, that Co5, Co6, and many other things, many other varieties from the different crops. They are doing marvelously at the farmers' field, and farmers, they are really getting benefited. So regarding this biotechnological interventions in forage crops, let me first introduce my state, where the Punjab stands as far as the forage crops are concerned. So we are roughly 9 lakh hectare area in Punjab under forage crops. We have two seasons there. Like we have a Rabi season and we have a Kharif season. We have a Rabi forage crops and we have a Kharif forage crops. In Rabi, we are growing mainly oats and barsim. Barsim, you know, it's a legume fodder. It is also known as the king of the fodder crops because of its high protein value, high biomass. So we have roughly 9 lakh hectare, or you can say 0.9 million hectare area. And Punjab is blessed. You might have heard the stories like we are amongst the top states as far as the dairy sector is concerned. It's totally mechanized. Even farmers, as I told, the silage entrepreneurship, silage making is another, you can say another event happening, which may help in bringing in another white revolution in the state. So that's one development I wanted to share. Silage making is going to be a hit all over country now. And again, we are taking a lead in that silage making. So for that, we are targeting oat silage and BN hybrid silage, maize silage, and sorghum silage. So four crops, they are found to be fit for silage. Number one is your maize. Maize is the number one crop in Punjab for Kharif season. It's best suited for silage. 1 lakh hectare area in Punjab under maize, spring maize. 15th February is the date of sowing. So 15th February onward, 1 lakh hectare area under this crop, just for 3 months. And they will be just harvesting at the dough stage, and the best silage they will get. And private companies, they are the real winners in this case. They are selling their seed hybrids for silage. None of the, only one or two companies, they have developed silage maize hybrids. Rest all they are selling is the your grain hybrids. So grain hybrids, they are being grown as a silage hybrids. So PAU has given two varieties I'll discuss. We have given two varieties, one J1006 40 years back, and still it is the national check. Look at the achievement of the breeder. 40 years back, a breeder gave a variety, it's a composite variety, J1006, and still it is ruling, along with the African tall, given by the breeder from MPKV Rahul. So only two varieties, none of the hybrid. Only just now the Adventa has given one hybrid, which is fodder hybrid. And very recently, last year, we gave another composite, J1007. So that's another composite variety. So you know the advantage of composites over the hybrids. Farmers need not to purchase the seed every year. So that's the additional advantage a composite is having. And plus it is giving at par yield to the hybrids. So there is no need to go for the hybrid. So that's another success story from the PAU, Punjab. So we are blessed to be, we can say we are meeting our GFY demand, means green fodder yield demand. 40 kg per animal per day is the requirement. This is the healthy animal, which is 300 kg weight, milk giving animal, that requires 40 kg per animal per day is the green fodder requirement. And we are providing 32 kg on papers. But in reality, we are providing more than that. Because nobody is generating data for fodder crops. That's a kind of dilemma for our crops. Nobody is willing to generate, no staff is there, no FDO is there. FDO, you know, it is fodder development officer. There might be agriculture development officer, agriculture officers, but there are no more now fodder development officer or forage officers in state government line departments. Nobody is there. So nobody is generating data. Only they are fudging the data here and there and creating. So we are surplus, or we can say we are meeting 40 kg per animal per day, depending upon the total population, livestock population in our state. Plus, we are having a plenty of dry matter. Plenty of. I was discussing with Dr. Ganesan yesterday that we are burning our paddy straw. You might have heard in newspapers that Punjab is blamed 10% farmers, paddy growing farmers. I just yesterday I came to know that 20 lakh hectare area under paddy in Tamil Nadu, but in Punjab it is 30 lakh hectare area under paddy. Paddy was never a crop of Punjab. So only during 80s, when the power supply, the power to the farmers, Punjab farmer is free. No charges are being levied. So that's good for farmers, but overall it's not that good. Means they are switching on and they forget irrigation. So paddy was never a crop of Punjab. We are now not growing any kind of millet in Punjab. Punjab is a land of Guru Nanak. Guru Nanak. So he was the first farmer on Punjab land who grew millets. Kodo millet. It started in Punjab by Guru Nanak. Okay? But now nobody is following his followings or disciplines given by, or the sermons given by the Guru Nanak. Nobody is now consuming millets. Nobody is growing millets. We are growing paddy and giving it to you people. We are giving our water to you in the form of paddy. 3,800 liters of water is required to grow 1 kg of rice, more or less 3,800. Somebody say 35, 36. So, an average 3,800 liters of water to grow 1 kg, and we are growing 30 lakh hectare area. So much water we are taken out. So many of we have 143 blocks in Punjab and 113 blocks, they are red zone, including Ludhiana. So we are harvesting our underground water. Okay. So with this background, keeping in mind in agriculture, Punjab agriculture, these are the agroclimatic zones of Punjab. Ludhiana is a central state, central district. We have 23 districts in Punjab, and Ludhiana is at the center of the state. Okay. So I discussed earlier also, we have roughly 81 lakh livestock population. These are our mandate crops. In Kharif, we have maize, pearl millet, BN hybrid. BN hybrid is catching up year by year. Even after coming here, I received 5-6 calls from the farmers. Sir, we need planting material of 5 acres, 6 acres, 3 acres. People, they are taking it as an entrepreneurship. This is a new trend I am observing at Punjab. Even the businessmen clan or the community, what they are doing is they have a land holding, they are planting BN hybrid to sell it as a planting material to the farmers. Getting my point? So they have taken it as a business, just selling the planting material, because it's a wonderful miracle crop, just like the coconut. Every part is being used, every part here is being used, and you plant it and it will go 10 years, 15 years, 20 years. In Punjab, farmers they have grown 30 years. The grandfather had grown, and now the grandchildren, they are harvesting. That's a scene in Punjab. It's such a lovely crop. Okay, the only thing is water guzzling. Even I have seen, if you don't give it too much water, it's as the result from today's studies from Kirnatha, that there will be a drop in the yield potential, but you won't see the mortality of the plants. That's another wonderful thing. Plants will not die because of the water stress. Once they get the moisture from the soil, they will start growing. Yes. Temperature in Punjab, another beauty of BN hybrid is, in Punjab, you see the temperature goes 4 degree, 3 degree for 20 days, 25 days. And during that chilling period, it has a power to tolerate that cold for so many days. So it has a winter dormancy fighting habit. It goes to dormant condition and then starts regrowing. That's another advantage of having this crop. Then in Rabi, basically I am, for the last 10 years, I am instrumental in bringing varieties in oats. In Punjab, farmers grow 1 lakh hectare of oats, Avena striva. And nowadays, in stores, in departmental stores, malls, you see there's a lot of maniac going like it's a wonder cereal. You know, it's rich in beta glucans. Beta glucans are there in Graminy family, but the content per consumption 100 gram, it is giving the best beta glucans. So beta glucans, they fight with your bad cholesterol and they make your heart healthy. And everybody wants to pay for the good health nowadays, particularly in our areas, northern states. The pockets are full of money and they want to get good cereals. People are there now, they are getting fed up of this consuming wheat and rice. They want some healthy food. I'm not saying that these are not healthy, but still, when you have a better option, so people want to go for a healthy choice. So oat fits there as a best breakfast cereal. So the world over, it has been promoted as a best breakfast cereal on the plate. European countries, Canada, America, Australia, they are consuming. The market is rising globally. But in India, not on to that scale. Many people tried, many companies tried, even Patanjali, Ramdev, he's trying hard, he's selling, giving free of cost packets, 100 gram with one bag of 10 kg wheat. But the problem is with our taste buds. It doesn't make a good taste. He tried to add some tomato sauce and everything to make it good, seasoned, tasty breakfast. But in Punjab, people are not like we wish to feed on our breakfast is full of ghee, butter, and prata. Nobody likes oats, but still we are instrumental in releasing the fodder varieties as well as the dual varieties, and now our effort is on grain oats. First grain variety we are targeting, and very recently, last year I gave first variety of the country at national level and at PAU state level. First dual variety of the country. So these are our thrust areas in PAU, you can say. When we are developing varieties for fodder, we are targeting high forage yielding varieties, high in GFY, DMY, quality including quality, biotic, abiotic stresses resistance, then high regeneration ability. So these are the thrust areas. Of course, the last one is very important for Punjab, like high seed yielding Barsim varieties. Barsim, there's a 2.5 lakh hectare area in Punjab. So highest acreage under one fodder crop. That's a Barsim because it gives 6 cuts. Farmer, they sow it in October, first week of October, and our varieties, they are late maturing varieties. PAU is ruling country as far as Barsim is concerned for the last 40, 50 years. Our BL series, BL, Barsim Ludhiana, they are number one. Because in the mid of June, when the temperature is soaring to 40, 45 degrees Celsius, even then it gives green fodder. And that with the 22% CP, that is a crude protein. So that's amazing because none of the kharif fodders, they have this much protein. So farmers, they are favoring this crop because you get multiple cuts in Rabi as well as in Kharif till June. So that's the beauty. And the cost of seed is 350 rupees a kg. And it sells like a hot cake. Within 2 hours in our Kisan Mela, first few hours, 2 or 3 hours, the whole seed is sold. So that's the story, success story of. And we are guiding our farmers that please come into the seed production entrepreneurship. Please. So they are coming up. As you have a Lucerne success stories in Coimbatore, Tamil Nadu, Lucerne is a main crop. So there, Barsim is a main crop. But farmers, they become greedy. They keep on cutting for fodder and don't leave it for seed. 8 kg per acre is required. 8 kg, which costs around 2,700 rupees to farmer. So at least we ask them leave a small piece of land. Don't take cut after your Onam or the 13th of April and leave it and take a seed. but many few are following our advice. That's a real challenge with us as a scientist, huh? To acquaint or to get the farmers along with us. What we are advising, not many are following. That's our lacuna, our failures. We develop a very good variety, and if it doesn't reach to farmers and farmers doesn't like it, what is the fun of developing such a variety? It remains in your CV. That's good for a breeder, but not for the society. So always try to be a good breeder, good in the sense that you give a variety that hit to the farmer. So look at the objectives of farmers. These are certain constraints that you might have read in your various courses, that forage crops, they are difficult to breed, they are heterozygous, heterogeneous, shy seed producers, and mixoploidy, alloploidy, autoploidy. Oat is an allohexaploid. Bean hybrid, you see, is a triploid. Okay, so there are many challenges. So they are difficult to handle, forage crops. Just to speak of a little achievement that we did in BAU, we could develop 74 varieties in the last 40 years, and 32 at national level and 42 at state level. And 27 varieties have been developed in the last 8 years. So 3.5, or you can see 3.25 varieties every year we are giving to the farmer. So the speed of breeding has increased. Particularly in barsim and oats, we are giving so many varieties through our Eclipse system. 12, 13 varieties in oats I have developed at national level. So this is the total germplasm holding with us. I started with oats 13 years back with 150 accessions. And now I have 1,000 plus accessions. So you are all students of GPB. So and you will be the future breeders. So your focus should be on accumulation of PGRs, plant genetic resources. Without that, you are nothing. How good you are at your breeding skills, all skills will be zero if you don't have the real wealth to work on. And the real wealth of a breeder is his germplasm or her germplasm. So your focus should be on the pre-breeding activities, on your accumulation of the germplasm. Make good contacts with the germplasm donors, means the institutes, your friends, your seniors, whosoever can give you some good germplasm. It's very difficult to get the germplasm. I'm working in Eclipse system. Our nodal agency is Jhansi, and I am very sorry to say that I couldn't get even a single line from the IGF or Jhansi. And I have said on big platforms also many a times that you are not distributing germplasm. In major other crops, it's not a problem, such a problem, like wheat and rice, they are distributing. Even their cement and your IRI, they are giving germplasm to the breeders because they know it's their mandate. But for forage crops, it's a lacuna. There is a dearth of germplasm sharing activities. So the accumulation of germplasm itself is a great achievement or activity in forage crops. Nobody will share. But TNAU, I am blessed. The very first perpurium lines that I got from Dr. Babu and Dr. Ganesan. So we are exchanging our material. So this is the best thing that breeders can do. You can share among yourself, at least. So this is the number of holdings in each crop. You see the number of crops we are working. We are 4 breeders there, and we are working on separate crops. I work on 3 crops: oats, bean hybrid, and third is a Guinea grass, which is another good crop for South India, for TN. So Guinea grass. So this is the 1,050 lines. This is last week's photograph at PAU fields. How we are maintaining our oats germplasm. Okay, now the story of oat will start. After giving you the viewpoint of holdings we have, now the story of oats will start. So the activity started after accumulating the germplasm, getting it from NBPGR also. And there is a research station in Shimla. They are also maintaining oats germplasm. NBPGR New Delhi, they are also handling 1500, 2000 germplasm lines. So I requested them 2, 3 times, and on the third time I got 320 good germplasm lines. So that becomes a good source material for the breeder as well as for students also. You work on that, you find the diversity and all those. So we started this work with one of our MSc student, my first MSc student in 2014. So that was a molecular characterization along with the morphoagronomic characterization of the oat accessions. And not much work was done through molecular characterization. It could be a normal thing in wheat, rice, pulses, oilseed, doing SSR markers. I did my PhD in 2003, completed 2003 my PhD, and I did in Brassica. And that time I used SSR. So you see here, after 11 years, we are using in forage crops, and it was a new kind of thing in forages, SSRs, microsatellites. Okay, so we could publish this paper in a very good journal of Iran. You may note it down. It could be very handy for you people when you are publishing your research. It's free of cost, although it takes around 8 months to 10 months for publishing. That is a JAST, JAST, the lower one, JAST. Journal of Agriculture, Science and Technology from Iran. So they took 8 months and they could publish our paper. Rating is nowadays is 7 point something. At that time it was 6 point something. That was a first molecular paper on oats, SSR molecular diversity analysis in oats, including Indian germplasm. Plus we could have another paper in range management agroforestry. This is another NAS rating 6 plus. So those who are working on forage crops, they must target this. This is a good paper, good journal. Although publishing time is a bit more, so more than 1 year sometimes they take, but it is worth waiting for students at least. 6 plus NAS rating free of cost, it's worth waiting. Then these are the pre-breeding activities that we are doing in oats for various traits, as Keithana did in BN hybrid, taking data recording for different traits. This is very handy. Sometimes it remains in the thesis and in the books and the papers. No, it should be in your notebooks. If you really want to be a good breeder, it should be in your hands. You should know my this particular accession is good for this very good reason. If I have 1,050 or 1,000 plus oats accessions, I must know my 675th accession I am having in my field is good for what? It is a high tillering, it's a high biomass, high CP, it's good in digestibility. For what I am maintaining my accession? Have you heard of mini core collections? Mini core collections. So you should have a working collection and then you should have a mini core collection. Some subset. 1,050 you can't evaluate every year. Okay. So you should have a 150 or around 200 lines coming out of the 1,050 lines, which you will be growing every year and focusing, which will be part and parcel of your every season activity. Yes, you can remove and add fresh entries every year. Because 1,000 entries or more than 1,000, 2,000 entries, you can't focus every time. Single breeder, you can't do. And everywhere, you know, the faculty strength is going down. The one crop, so many accessions, single breeder, because he has to take classes and everything. Your Dr. Sab is doing so many activities daily. So it's better, the focused approach. Your mini core collection should be there. What should be there? Mini core collection should be there. So this is a recent pictures. We have started making crosses and we are utilizing services of students. They learn. Okay. Whatever practical or a theory courses I take 10 marks assignment to students. I give 5 female lines and 5 male lines. You give me combinations. One combination, 1 mark. Isn't it good? because this is the only science where you create. Other sciences, they work on the material created by a breeder. Dr. Saab is creating BN hybrid. Now the job of the entomologist, agronomist, soil scientist starts. Once you give a new variety as a breeder, only then the job of agronomist will start. He will start looking whether the Dr. Saab hybrid needs more fertilizer doses, less doses, how much should be the irrigation requirement, any seed rate should be increased or decreased. What is new pest coming? If Dr. Saab gives a new variety which is resistant to all the diseases, all insect pests, it means your pathologists and entomologists now are free because variety is so good. Inherited traits are there, inherited genes are there. So that's the beauty of non-breeding. So you are blessed, you are a breeders, budding breeders. Okay? So just a glimpse of how many breeding material we are generating and handling every year. So there was a QRT last year. QRT is your 5 years review. So there I presented this slide. So this is a 5-year review you see there, 16 to 21. So we are handling number of progenies because it's a self-pollinated crop. So you are having F1, F2, F3, F4, F5. The I can recall a very good this story. During 2013, I was presenting this table in 2013. So there were, I made 100 plus crosses in oats. 100 plus crosses. So everybody, including the chairman of that session, was like, how you can make 100 plus crosses in oats? Because the others were making 9 crosses, 8 crosses, 6 crosses. One center made 2 crosses. So means what else breeder has to do? You will make crosses, only then your job starts. No? Until unless you make the gametes to join to create a recombinant, how will you work? You can't work with the introduction now, you can't work with the selection now. Here in self-pollinated crops, your job starts only once you make a good combination. So these are some of the material. On the right side, you see, this is a this year photograph. Till now, we have in the country oats which is giving 2 cuts only. So this is we are targeting first variety of the country which will give 3 cuts under ideal conditions, normal conditions, not like sowing earlier and like that. Under normal conditions, normal fertilizer, normal irrigation conditions. So we are targeting. So first cut is to be taken at 70 days after sowing, like we are sowing in 10th of October. So on 10th of December, we will be taking first cut. After 40 days, second cut, and then till maturity. Maturity means when milk stage comes. Milk stage in the grain. We call it a dough stage. Then it will be harvested. So that gives, actually, there our target is to compete with the Barsim. Barsim gives 5 to 6 cuts. Farmers, they ask more cuts. They don't want a single cut. They want multi-cuts, particularly in Punjab. Because that's all machinery driven. They don't do it with the hands. Manual is not done. We have a machines, fodder harvesters. So they run it within half an hour or 20 minutes, 1 acre is cut. So after that, they apply fertilizer and irrigation, and the second cut is ready after a month. So this is what they want. So we are targeting our regional farmers. That is my first variety in 2014. And it is going very good. It is more than 6 feet. I am 6 feet. It is 6 feet, 6.5 feet. Nobody saw in my state also this tall oats. They were saying, sir, is it oat or a pearl millet? Okay. So after this, what new initiative we have taken in the last 4 or 5 years? In 2017, I submitted one project on intergeneric hybridization. As you saw today, that was an interspecific hybridization that Kirtana attempted. So that was intergeneric hybridization. You might have heard of wheat into maize. In your courses, you are taught wide hybridization. And they give example of wheat into rye. That is an old example. And BAU is lead in country in wheat into maize system, haploid production system. You know, speed breeding, accelerated breeding. What you can do with the haploid, like we take around 10 to 12 years in developing a self-pollinated variety through pedigree method. But through the double haploid production, you can minimize the number of years from 10 to 12 to only 3. You will be cutting down the number of years. It means every third year or fourth year, you will be ready with the new variety. Yes. Same thing we tried in oat into maize, but with not much success, with a little success. And now we have one of my another MSc student. She has again started working on same problem. Okay. So that is oat into maize. Addition lines. What happens in wheat into maize? In wheat into maize, your maize chromosomes, they get eliminated. That's an elimination, chromosome elimination technique nature has provided. Only the 21 chromosomes of wheat. Wheat has allohexaploid genomic constitution. 21 chromosomes from wheat comes, but 10 of the maize doesn't come in F1. So your F1 is haploid for 21 chromosomes coming from wheat. So those 21, they are doubled and we get 42. And what's the advantage there? You are fixing all the allelic combinations. So it means your 100% homozygosity is achieved, which is achieved in a normal pedigree method during 8 to 9 years. You go up to F7, na? F1, F2, F3, F4, F5, F6, F7. So here you are achieving the same homozygosity in 2-3 years. And we are using facilities as Kelong. Kelong is one place in Himachal Pradesh where we're taking this off-season nursery. So we are using 2 seasons in a year. So you see, in 1.5 or 2 years, we are ready with a variety in Punjab for wheat. Very good lab there in PAU. It's a haploid lab. We call it a DH lab, double haploid lab. So lots of funds have been received from ICR, DST, DBT. So it's a very good lab, a worth seeing lab. So oat is a C3 as wheat, rice, they are C3. The idea was to see if a maize chromosome retains in a oat, it retains. We get addition lines. System is not similar, it is dissimilar to the wheat into maize. In oat into maize, we get maize chromosomes retaining in the oat. So there is a frequency, means 1 chromosome, 2 chromosome, or they can be haploid. So you get lots of genetic starts. You get 1 chromosome retaining, monosomic, 2 chromosomes retaining, disomics, or if none is retaining, you get a haploid. So we were targeting the addition lines. If there is an addition line, how the C4 physiology is behaving in the C3 plants. That was a target in developing oat maize addition lines. Okay. So these were the objectives I discussed. This was a work plan. This was a 3-year project with 30 lakhs. So this is some of the pics. Embryo rescue. We did actually the problem was, we thought earlier that the same system will work, kind of your embryo rescue technique, the media and all those things, but it didn't work. Same means the similar that we are doing in wheat into maize, it will work in the oat into maize. The media didn't work for oat into maize. So we spent 1.5 or 2 years in standardizing those protocols. And from that, we could get one good paper. The standardization of the embryo rescue technique in oat into maize system. We made 6,000 pollinations in a season. 6,000 pollinations means 6,000 spikelets were opened, masculated hand masculation was done, and hand pollination. This was a daunting task. 6,000 spikelets to be pollinated in a season. Of course, done by the research fellow, one research fellow and one MSc student. And this was the publication, not that much good publication. So attempt to produce odd haploid using odd maize hybridization technique. International Journal of Pure and Applied. Name is international, but journal is not international. Rating is 5.8 something. So this is present. Nowadays, what we are doing in our fields, crop is in our field. What my students are doing, what I am doing. We are exploring in vitro, in vivo, double haploid techniques in course. So you know haploid, I told you how haploids, they help us in faster breeding, speeder breeding, accelerated breeding. We are targeting one locus. You see this locus? This is known as ZMPLA1 locus, also known as not like that, NLD locus. And it has been aligned in many other crops. You know what is paralogs, orthologs, homologs, ortholog, paralogs, gene sequences. So we are targeting the ortholog sequence in oat because it has been sequenced in other crops. Okay? Ortholog gene sequences are the gene sequences which have been conserved by dysentery. You know, there was only one ancestor which gave rise to all the Graminy family crops. We have maize, we have wheat, rice, barley, oats. They have all come from the one single parent, which was having this ZNM material. We are looking for that particular gene locus in our today's cultivated oats, through which, if it is there, through which we can develop haploid. Means then there may not be that much requirement of doing ode into maize. Simply by knocking out that gene, we can get a haploid gene. Okay? So this is one another thing that we are targeting. So we are improving response of oat maize crosses, and we are in silico studies. In silico means we will be taking help of bioinformatics, and we will be looking for the sequence because oat genome has been sequenced. This is another achievement at the global level. We never expected because of the least importance being given at the global level also. But only one single group is working of 15 scientists in oats globally, but they could come up with a very good saturated oat genome map. So this is handy for us. Looking at the genomic constitution, genome sequences available, we can now search that sequence in our genome, whether it is there or not. Okay, so this can tell us on which chromosome that locus is present. So targeting that locus, we can go ahead with the in silico studies. Okay. So our expected outcome will be here in this case that there will be the improvement in the oat into maize double haploid system, and there will be generation of the in vivo inducer. So that will act as an in vivo inducer that will induce the haploid system. Okay. Then another thing that we are targeting, as I told you, that oat is gaining importance as a breakfast cereal because of its remarkable chemical constituent, which is nothing but a soluble carbohydrate. That's a beta glucan. Okay, we call it a beta 1,6 glucan. So this really binds with your bad cholesterol, to which we call the LDL, low density lipoproteins. Okay, so this CHD, this coronary heart disease is coming up very heavily, particularly in North Indian population. Punjabis, we are getting, every other house is having your grandfather, your parents suffering from CHD. Many of my colleagues, they look thin, everything is fine. But the other day we get a report, sir, they are getting stents, artery stents. They're putting stents because of atherosclerosis. Huh? Cholesterol get deposited in your arteries and veins and causing problem. So this can be handy. Only 40 grams is to be consumed daily. And reports are, international reports are there. After 3 months of 40 gram consumption daily, your LDL is reduced by 30%. It's a big number, 40% reduction in the LDL. So the idea was to give a beta glucan rich oat variety. Whatever oats we are consuming, either it is coming from outside or it is the fodder oat seed that you are consuming as a oat. It means there is no variety, such variety present, which can be consumed for the cereal. So the objective was to develop a variety at a national level, which will suitably or exclusively to be grown by the farmers as a breakfast cereal variety. Okay. So this was a tie-up with the private company, GSK, GlaxoSmithKline. So this I told you that oat beta glucan. This PPT will be here and it will be shared with you people if you really want. Dr. will share. So these were our objectives. Whatever germplasm we acquired, you just characterize using the microsatellite markers to develop the mapping populations. Biochemically, we will be characterizing what are the lines which are having the low beta glucan and high beta glucan, crossing them and developing the mapping population, F2, F3 mapping populations, and to identify the QTL for the beta glucan. There was only single report from the European country, and it was the first report in India, QTL mapping in for beta glucan. So we could find our own lines, released varieties, both. OL stands for Oats Ludhiana, as you have Coimbatore CO lines, CO varieties. So PAU varieties, fodder varieties are named as OL, Oats Ludhiana, OL10 and OL11. OL10 was found to have close to 6%, which is an international standard of beta glucan. Now they have reduced it is roughly 4.5 to 5% beta glucan, but it was having 5.8% beta glucan. And another my variety OL11 released. What's the beauty of the OL11, sir? It has been released for your state as OL1760. This is the first variety to be released for South Zone. Single cut oat variety OL1760. We got it released in our state also as OL11. But it was found to have a low beta glucan because it was never bred for beta glucan. So luckily we got parents OL10 and OL11. We developed the mapping population. These were the set of markers that we used. More than 250 SSR markers we used. 75 were polymorphic. Number is low, 75. But whatever resources we have now, nobody is willing to fund forage crops. That's a tragedy with us. Funding agencies, they are happy to fund big crops, wheat, rice, pulses, oilseed, cotton. That's a tragedy with us. We get limited funds. There's a tussle going on. Ministry of Agriculture and Ministry of Dairy. They say these are your crop. They say no, this is your crop. So when funding comes, the tussle is going on. It's not they are means we are working for dairy, na? So they say you should fund. Why we should fund? So that's in tussle. Scientists they are suffering and the community is suffering. So we could map few QTLs on linkage map. These two QTLs, QTL cartographer, it mapped two QTLs using the composite interval mapping approach. You know, there is a simple interval mapping approach and composite interval mapping. So we used composite interval mapping approach and we could map two QTLs. So these were the publications out of that project. One very good publication we could have, that was an International Journal of Elsevier, 9.8 rating, this one, LWT, Food Science and Technology. Then 6.8, Indian Journal of Genetics and Plant Breeding. And very recently, we could publish another review paper on nutritional benefits of oats. It's a multi-institutional project, and crop and pasture science, 9.8. Okay, so this tells the story that forage crops, they can be high yielding to a student as well as to a scientist. So don't get afraid or refrain yourself from coming to forage groups. They are waiting for you. So this is the first variety of the country which is released for dual. Dual here makes a sense, means it gives two things. Means it will give you green fodder. After taking the green fodder, 70 days after sowing, you leave the crop and then you take the green. It's not in our case, sir. In Bajra, it is dual is a misnomer. Misnomer means we say dual Bajra, dual per millet. It doesn't sound like taking Bajra first for grain fodder and then leaving for grain. No. Dual in that case is either fodder or grain. Means you take this variety, it is as good as for fodder as for grain. But here it is a real sense is a dual. You cut it for a grain fodder, farmer will get a fodder also for his livestock, and then he can sell that grain, which is rich in 6% beta glucan, along with the other nutritional parameters. It's a kind of biofortified oats. We refrained from using the word biofortified, rather we used the term dual oat variety. So it was released at national level as OL 1876-2 for Northeast June. 5 states are there in Northeast in our active system, and Punjab as OL 16. So I started from OL 10, then 11, 12, 13, 14, 15, 16. Next year I will be ready with the OL 17. So just showing this was released at national level. Then another, I'll take just 10 minutes, not more than that. Okay. So another study that we could take up, another project that is again on QTL. This is a Helminthosporum leaf blight. It's also known as a Pyrenophora leaf spot. It's a major disease in northern states. Sometimes my own variety, they couldn't get released at national level because of this menace, this disease. And it causes damage up to 30-40%. Blight. It reduces the foliage quality also, because when the leaves will be covered with blight, so everything will be suffering. Okay, so we targeted the QTL mapping in, but this particular disease. This was one of my PhD student. He just completed last week. Viva happened last week, and very good publications could come out of this project. And we are targeting another very good publication, because this is the first report. There was only one report way back, and we are reporting. And also we are giving gene postulation. Means how many genes are involved in. Nothing was known regarding its gene postulation, whether major gene is there, minor gene is there, or some inhibitory gene action is there, or masking gene is there. So we could postulate through our studies that there was a kind of masking gene action through the ratios that we got. Mendelian ratios, you know, na? Non-Mendelian ratios. So this was how it was done. The whole germplasm, 96 germplasm lines were screened. Then R and S donors were selected. There was no known R and S. Generally, in your crops, there may be a well-known resistant donor, well-known susceptible donor. It is not there in our crops. So first we have to identify, after 2 years of application of this inoculations, artificial inoculations with the pathogen, we need to find out which is the R parent and which is the S parent. Then making crosses and then getting the mapping populations. So it was a daunting task amid this corona period. So it was a challenge for student as well. So this is how the progression of disease looks like. How the disease progresses. covers the whole of the leaf and then whole of the plant is covered with the pathogen. So we could get out of the, now you see how that acquiring the germplasm from the NBPGR helped here. You know what is EC stands for? EC? Exotic collection. IC for indigenous collection. So you see the EC collections I got from NBPGR, both they were handy in our this study. One EC was resistant or MR, moderately resistant, and other was susceptible, 22 and 70. So we could use those lines in developing our mapping population. So this is a frequency distribution of different parents. Then how many crosses we made, means how many panicles and how many, this is how the crossing was made. This is student, Hilly. He's from Karnataka. Then we use the facilities, off-season facilities at Kalon. I told you, in Himachal Pradesh. It is close to Leh Ladakh, Kalon. Today's temperature is -2 degrees. Today's, I just checked in the morning. Here we postulated, through the ratios we got, that there could be a masking gene action. Okay? Then we did our molecular analysis. Different markers having high peak value, they were selected. And we did the QTL mapping. One minor QTL and one major QTL was found. Okay? So we concluded that this is our resistance source. So now anybody who now wants, so this is the outcome. We identified this is a resistance source. So if Dr. Saab wants to work here, we can then give our resistance source, use it as a resistance source. Okay, you want to screen population or you want to screen germplasm for this disease in your area, we can provide you resistant and susceptible sources. You need to have susceptible source no? For using it as a control over screening. So earlier it was not available. So we have generated both resistant and susceptible donors. Populations are available, so even populations can be given. So we could publish this first paper in 5.4 NAS. And another I missed that was published in CRC, the Serial Research Communication. That is another major paper came from his student study on this HLB, Helminthosporium leaf blight, in the Serial Research Communication. Then what we are doing just in 5 minutes, because BN hybrid is your region's crop, you are doing excellent, CO5, CO6 is doing excellent, and we have just saw Kirtani has done marvelous work in BN hybrid. So we are also, because I told you BN hybrid is gaining importance in Punjab. So in BN hybrid also we are doing these kind of studies like identifying the superior parents for different traits, and then we are crossing the crossing is very difficult there. Actually, flowering comes in month of November and or December, and by that time bajra is not there. Bajra is a kharif crop there, na? It gets over by the October. In October, we don't have flowering in purpureum, napier grass. So we need to make use of controlled conditions. We grow late bajra in protected. This is glass houses or poly houses, and we take pollens from outside and then make crosses. But still, we are managing. Then new initiative was taken, like you have seen, we are distributing it to farmers. It becomes very difficult to transfer. Also, farmers, it's very difficult to take it to far off places. How to take some bulky material. So we tried Colchi diplodization, doubling the 3n to 3x to 6x. And let us see if there is any seed set or not. We could double the chromosomes, but couldn't get seed. I don't know whether our climate is playing some problem, spoil spot or something. But if a same genotype, we can bring it here and we can see what the climate, because the photoperiodism and all those, they are regulating this. So that is to be seen. Clones are available with me, where it is cytogenetically as well as using the scanning electron microscope. It shows that the stomatal size and frequency has increased. So in polyploids, as you go higher in the polyploidy level, your stomatal size increases. This is what the literature says. Number of stomata reduces, the size of the stomata increases. So also the trichome density, that is hairiness. So as we go higher the polyploidy, your trichome density reduces. So these two parameters along with the cytogenetic investigation could reveal that there were clones having doubled the chromosomes. And we could publish in good journal. This is the stomatal sizes. You can see. So big stomatal size. Indian Journal of Genetics and Plant Breeding, 6.5 or 6. A student went to Australia after doing this, funded a fellowship. So another publication is stuck. Okay, so when number of students are more, so naturally the one who is with you available all the time. So your focus is on that publication. Otherwise, we could have targeted more publication. It was a first report from India side that quality diploidization is successful. Then I talked about the soma clonal variations in BN hybrid. As Dr. Saab told, there is a heterogeneous nature of the purpurium. They are not fixed. Nobody has fixed them. Means they are not a kind of inbred lines as we have in Bajra. We have A and ABR line system. It's not like that. It's a heterozygous line. Every pollen is different. So every F1 seed that you get is different. Although it is F1 seed, but it is not fixed. It is different because of the heterozygous nature of the purpurium. Okay. But the availability of the purpurium is very, genetic diversity is narrow. I think the same lines they are roaming around the whole country. They have given their own names. Like we have a FD here, the same FD when reaches to me, I give Ludhiana number. So same entry may be having different nomination. So that makes a problem. So I believe there are not more than 40, 50 or 60 lines roaming around the whole country. You see the genetic base. Only 60 or at the most 100 lines only. And you are targeting only those 100 lines. You see the number of lines she has used. How many, Ethan? 5? 5 lines per purium? 5 lines. Per purium 15. 3 only. So she selected maybe morphologically that these looks good for what it's. So see the genetic base. So the idea was to improve, to broaden the genetic base, so that we have more available variation. So with that approach, we went for somaclonal variation. So again, there was no protocol established. Means what will be the shooting media, what will be the callousing media, what will be the rooting media. Nothing was available for our crop. We borrowed it from sugarcane. 50% worked for us and 50% we made our own efforts. And we could establish our own media first time that if you really want to go for tissue culturing of BN hybrid, you can go with this media. Establishing the tissue culture protocol in Bajra Napier hybrid. Okay, and we could get 3-4 soma clones also. I'll show you. So this is how we went for callousing, then shooting, then rooting. You see here, roots are coming. Then establishment in the field itself. That all was done during the Corona period. And then the molecular characterization. How would you say? This is a soma clone. This could be a field variation. You're getting more tillers. It could be a labor problem. You put more fertilizer into that clone, more water given to that clone, it is giving more tiller. We confirmed it with the marker. Which marker? Purpurium, chromosome specific marker. Okay? You see here, different bending pattern. You see here. We are getting 3 bands here, and rest all we are getting multiple bands. So this is how it looks like in the field. So they are in the field, and we are targeting the ETOC journal, plant tissue organ culture. The rating is 11 or 12. So earlier it was charging $1,500, but from the 1st of January, all Springer journals are free. So you may target. For India, Springer has waived off the application processing charges. So it is very good for us that earlier you are doing very good work, but because of the charges, you never target high-rated journals. So Springer has waived off now the this article processing charges for India. So these are some somaclonal variants we have identified. Although here for the thesis, lots of somaclonal variants are there, but corroborating your morphoagronomic and field performance, how they really look like and how they look on the gel. So 3, 4, they are very extraordinary. Really, they are the variants. So we are targeting publication as well as the registration of this genetic stock as a this is a equivalent to variety release, and you see the work of the student going up to that level. Means we are going to register that clone or a somaclonal variant as a genetic stock. Okay. So this is the overall conclusion what I discussed with you in oats and BN hybrid, because I told you my success stories, which are really coming out of my lab, my fields, not what is happening here and there. They can be theoretical things that you are studying in your, or you are being taught by your teachers during your theory programs, your theory lectures and practicals. But these are the real things that we are doing and we have done with our hands from our labs and publications coming from our labs. Not that good, but still for forages, it is good. To bring forages in limelight, it is good if our publications are going. So I would say your biotechnological approaches, they are very handy tools. Even now the CRISPR-Cas is coming like it's a mind-blowing tool. Everybody is using CRISPR-Cas in wheat, rice, and any other crop. So we are writing one project and we are just going to submit next month SERB, SERB. So we are submitting to SERB one project on CRISPR-Cas in oats. So if wheat and rice can do, we can also do CRISPR-Cas in. What they yield, that is different. Okay, but to be in a race, you should be ready. So that is. Thank you so much for your patience listening. Any queries? Any queries? For your educational tour, come to PAU. Or you make a tour to northern states. Okay? Make a plan with your teachers and do come to PAU. We'll welcome you. We show all the labs. We have a very good biotechnology division there. School of Agricultural Biotechnology. Very recently, it is funded by 27 crores by DBT, Department of Biotechnology. New building is coming up. And 19 RFs have been appointed. I was just telling Kirtani, she is not ready to leave Tamil Nadu. So there is a lot of opportunity for you students to come and join there. Okay? Yes, your queries.
Sir, double haploid and single sequence method, both fast homozygous line give. Then why use a double haploid?
That is accelerated breeding. Single seed descent method again will be taking 8, 9, 10 years, 12 years.
No, sir. Off season or greenhouse also use in single
That is
seed.
good, but if we are using off season in double haploid also, no? So we are ready in 2 years.
But double haploid is very sophisticated method. costly than single seed descent method?
That is one means two side of the same coin. Okay, the coin is same. It depends on you. You want to, I won't say waste your years. If you want to spend good number of years in developing a variety and you want to spend just 2 years. So actually in staple crops, no, wheat and rice, you see, there's a race going on between, you can't imagine the level of competition between the IARI, IIWBR, Indian Institute of Wheat Barley Research, Karnal, and PA. Who comes up first with the leading variety for North June? Okay, so for that sense, we really wanted this kind of system with which we are now ready, because they are using, IARI is already using. So we have to have that system. Moreover, your chances of committing errors, they are low in your, and plus you are getting 100% homozygosity, complete, because that will double all the locus, now. There you will not achieve 100% homozygosity after so many years. That's an added advantage. But the prime advantage is you are saving time. You are correct, but now we are we have developed kind of system where you don't much require so hi-fi technology. Only you need chemicals for tissue culture, for the embryo rescue. Colchicine is not that costly. Okay, if you are purchasing in bulk, so it is already available. Plus the concentration you are using for doubling the chromosome is very meager. So 0.1% or sometimes it is 0.2%. It's not that. 1 gram colchicine costs 4,500 rupees. It's not that costly. Yes, you need expertise. Their DPLs are doing, our daily paid laborers, they are doing embryo rescue. Yes. For the last 5-7 years, we have made them so much experience. They are doing regularly. Students are doing outside pollinations and everything. We have given the tiller culture method. What we do is we emasculate, pollinate with the maize pollen, and after 21 days, we cut the tiller and brought it into our lab, put it in under the chilling conditions, refrigerated, and there all things they go on. Okay, so initial setup was difficult, but now it is a routine thing. Yes, please. 1 mark. Those who ask question. Yes, it is working.
So I have doubt something related to not exactly this previous PhD.
You keep it
study.
here.
So a topic came about, you asked a question about CMS lines in Bajra. So I have one doubt. As of now, current is A5 cytoplasm used in Bajra?
Huh?
A5, is the A5 used now in Bajra?
Yeah. That's how we are using A5 now. Well,
Yes,
up to we have
actually,
A7 also, violation also, Garo also.
I was not aware of current what is happening. That's why. Our restorer lines, I've heard like Ikrizat have restorer lines were not available. Restorer was not available, especially for A5 and all. So Ikrizat was developing. They took restorer from A1. So it's that was my doubt, whether is it available.
That's
Is
now
it
restorers for, I think.
Actually, for a grind development, we are looking for A5 and the A6, even the violation based A1 to A5, and another 2 more male sterile systems are there. But the majority of the restorer lines we couldn't find difficult to get in the A5 based CMS. But for as far as forage is concerned, once we make some crosses, the A5 based lines also can be used, because the directly crossed seeds will be sown and we'll be making selection in the F1. So that won't be a problem. But only in F1, there won't be any seed set when we use the different set of CMS lines. But for a forage purpose, we can use any type of CMS lines. So there is no issue about that.
Problem with the ICRI set is they don't give you B line. Whenever we ask germplasm, they give a set of A and R lines. So after 1 year or one season, you are again handicapped. You can't use A line, you don't have seed of A line. Na? For the seed of A line, you need to have a B line also. So they never give B line. They only give A and R line. You develop hybrid, you are very happy. Hybrid is performing better, but you have again go back to them, telling them that their policy, it's an international institute. You have to tell them this is a hybrid that we have developed. It is performing excellent. It's overpowered, out yielded all the existing hybrids. Please give us. So there will be a this MTA, material transfer agreement. And it's means they will give you, but it's a daunting task of getting a B line also.
But we are having, sir. So now they are giving the even the
Giving the B line.
A5 based lines also, but getting the very good.
So we were speaking about that colchicine induced exaploid in Napier grass and bajra. Actually, this was discovered in sorghum, right? So is there any mainly this is spoken with respect to sorghum, this colchicine induced polyploidization. So in PAU, like also work is going on sorghum. So what's
No,
the status?
in sorghum we are
terms of-
not using this. This is some another sorghum breeder is there. There we are targeting Sudex hybrid. You know Sudex? Sorghum into Sudan grass. Sorghum bicolor, and another species we are using is a Sorghum sudanensis, Sudan grass. Okay, so it is an interspecific hybrid. The first interspecific hybrid in sorghum was given by PAU in the world. That was PSC1, Punjab Sudex Cherry. Cherry is a vernacular name of sorghum. 1, PSC1. And still its seed is being produced by the NSC, National Seed Corporation. So way back, all the ABR lines were given to NSC, free of cost. And every year they are selling worth of crores of rupees seed to farmer, PSC1. And 2 years back, we have released PSC4, Punjab Sudex Cherry 4. So you are right that we can use this quality diploidization method, but we are actually these are the different tools available with a breeder. Whether you want to go with the normal hybridization method, you are not getting much achievements, much success in normal hybridization. You want to tap the WCR, means your wild crop relatives. To tap the wild crop relatives across the gene pools like secondary tertiary gene pools, you need to have different tools. Then you can, of course, go for the polyploidy breeding as we did. You can go for the EMS, MMS, that is your methyl sulfonate, like your mutants. So there's always, I was telling you that PAU is leading in Barsim. All Barsim varieties except 2, what PAU has released now, till now, they all bred with the mutation breeding. Whatever, there's one variety from EAU that is a BL10, Barsim Ludhiana 10. Farmers, they are mad about this variety. We have given BL42, BL43, BL44, BL45, 4 varieties after BL10 with the gap of 22 years. 22 years ago, BL10 was released. But again, farmer is coming and asking for BL10. That's our drawback. We take it on our shoulders. We are not that competent. We can't make him realize, please go with the BL44, BL40. No, sir. Please, if you have BL10, then please give me. They're always asking BL10. The only thing with BL10 is it gives last cup up to end June. Rest all varieties, they finish before 15th of June. Just with the 15 days of higher, bigger maturity period, longer duration, they are looking for BL10. And in black market, it is being sold at 500 rupees a kg. Black marketing. Because naturally, if it is a late maturing, flowering will be late, and by that time the pre-monsoon shower comes and the seed production is very less. We generally say 2 quintals to 3 quintals seed production in Barsim from 1 acre, and it gives only 1 quintals, half the seed production. So still it is very popular. So here, live example where mutation breeding has been extensively used in developing varieties. So forage crops, they are using each and every tools, be it polyplorization, mutation building, wide hybridization. There are so many examples. Okay.
So for mapping, we need two extremities usually, but we were speaking about accessions where we are having very less 60 or 70. So we get false results, right? If we are having very small
No,
accession.
for that you need to be doubly sure. Either you go with the multi locations and multi year. Okay. Before you start developing your mapping populations, either it should be a standard donor that throughout the year it is established that this is a resistant and this is a susceptible. But once you don't have, the approach left is, because for students we can't wait for that long that we'll keep testing whether it is a susceptible or resistant. So for 2 years we did the inoculations both naturally as well as naturally means dense planting also we tried, because in dense planting giving extra nitrogen more flourish is the disease. Okay, so we tried all methods and then we could arrive at a conclusion that these are the two accessions which are as for now available with us. There could be any other source, but what is available with us out of those, this is the R and this is the MR, not R, MR and S. Okay.
Yes, sir. Thank you.
In one of the slides, you told you have been using that the cluster beans. What purpose that cluster bean is used?
Sir, cluster bean was grown as a fodder crop in some of our districts in Punjab. Like there is a district known as Bathinda. In many movies, Bathinda name is Bathinda. So in that area and some other 2-3 districts where farmers they were growing, because this is the only pulse crop available with the Kharif season. Cowpea and cluster bean. We call it guara. It's a cluster bean as a fodder crop. But last 4-5 years back, there was a huge demand for a cluster bean because of its guar gum. That the gum that is extracted from the guar cluster bean. So the crop was sold in the Indian grain markets at 40,000 rupees a quintal, which is now nobody is ready to take 4,000 rupees a quintal. It was sold by at the rupees of MSP, not MSP, but the market price was 40,000 rupees a quintal for the Semopsis. That's a guar.
Sir, I have a doubt. Sir, you have told about wide hybridization in oat and maize. Help us to know about the mechanism of conversion of C3 plant into C4. Like, can you just elaborate this? I didn't got.
Actually, please visit it. We were targeting because C4 we have a bundle sheath mechanism and all those their mechanism like your sugarcane. It's a C4. They are means they are known to be high energy efficient crops with the whatever available solar energy is with them. They can convert that much energy to make much of the carbohydrates. This is what the sorghum and sugarcane they does. Okay.
Yes, sir.
So we wish to transfer some of the C4 traits into C3.
Sir, in oat and maize cross, the maize chromosome get eliminated, right?
It is retained.
Retained.
It is retained. We have 2 addition lines developed. Okay. But internationally, there are 10 chromosomes coming from maize. For 7 of the chromosomes, addition lines are there. 3 chromosomes, they are known as mischievous chromosomes. They don't enter into the, or they don't retain into the this harmony. This is a disharmony between the genomes. So that is known as a genomic disharmony between the oat genome and the maize genome. So 3 chromosomes, they are not coming into the oat background. Only the 7 they are coming, and there are 7 different separate addition lines are available. We have 2 addition lines for chromosome number 3 and 7. But you were asking what C4 up to that level we have not yet, because our focus is shifted towards the development of the DH. Rather looking at the C4 expression in the C3 background, focus now is to develop DH, double haploids. Please, from back side. Come forward. Come, please. No, only two objectives. Main two objectives were there. One is how the maize chromosome will be behaving from the angle of your C4 expressing in C3. And the second was to extract the haploid and then doubling to the double haploid. So these were the two major objectives. This is very, and particularly in wheat into maize, so very stable system. It's not like that your DH will break down or something like that, or instability at genomic level, or there is a cytonuclear, any kind of disarmament. Nothing is like that. It's a very stable system. And PAU is using for the last 10, 12 years. And now the Palampur University, CSK, HPKV, their new vice chancellor, he is a biotechnologist. So they are using not wheat into maize, but they are using wheat into Imperata cylindrica. Imperata cylindrica. That's a weed. So they're using that weed in spite of, despite using this maize, because maize pollen getting in Rabi season, cold condition is very difficult. Maize is a kharif crop, you know, summer season crop. So they were not getting pollens there. So they tried to have another species or another genera. So they found that Imperata cylindrica is working very well, and they are also doing excellent work in this DH.
Good afternoon, sir. Sir, one question. Out of curiosity, I'm asking, not probably not a
Good.
question.
Curiosity, what makes the
Yes,
science
sir.
working?
Sir, actually, you have told about OL11 series, no, sir, which is suitable for the south zone of
Yeah.
currently. And my question is like, what could be the possible objective that could be targeted so as to achieve so as to come out with a novel achievement?
Actually means cultivating oats here.
Yes, sir, for this zone.
This will be a new crop, means you're adding something new to the basket of your dairy farmers. They are growing BN hybrids. They are growing, actually oats, you can't compare the qualities of BN hybrid and oats. Oat is far superior than BN hybrid. Be it a palatability. Palatability is more than 62%, 65%. Your CP is higher. CP in BN hybrid in Punjab conditions, the CP is 6 to 7%. But in oats, CP is 13 to 14%, double. So you are getting double the quality. Plus palatability is higher. Okay, so you are giving new crop to the farmers. Plus silage, you can farmers they can go with the silage also. So that's a advantage of growing oats here. Oat will work as a single cut here. So all the trials in active fodder crop, whatever oat is being grown is towards your hilly areas. We have a trial in Ooty. The data regularly comes from your Ooty place. So there's one scientist working there through WhatsApp. So trials come to him and then goes to Ooty and the data comes to us. So through that data and through your Kerala data and one Karnataka data. So 3-4 states, but Telangana is there, Andhra is there. So 4-5 states through that data, that OL11, which is 1760, OL1760, that was released for your state also. So if Dr. Saab is willing, I am happy to give seed because I want my varieties need to be popularized. If it is for your zone, it needs to be. But very good policy at PAU, you will be amazed to see if Dr. Saab develop CO5 or CO6 for my zone. So I have to take a release proposal to my university research evaluation committee for its state release because CO6 or CO5 is released for Northwest Zone and Punjab is one of the location, Ludhiana. So I will take a proposal, a release proposal to my research committee that this hybrid released by Dr. Ganesan has been found suitable for Ludhiana or Punjab at national level trials. So let us conduct 2 years or 1 year trial, see the suitability. If it is good, then release it for state. But in many other states, nothing is happening. So with that, what is happening? I am giving a variety. My maximum oat varieties, they have been released at national level, be it a central zone, be it a northeast zone, this 1876-2, that is OL 16, first dual variety. This is released for Northeast zone. Who bothers? Northeast zone, we have a Imphal, we have a Assam, Jorhat is there, you have the Jharkhand, Raipur, West Bengal. So till now, nobody asked, sir, give 1 kg sealed varieties released for our state. And that's a tragedy. Because varieties for your state, it means in your trials, this performed outstandingly. Then the checks. So if it is performing outstandingly, at least we give a go. Again, 1 or 2 years trial you can conduct. And if it is performing better, we should go ahead with the variety. Any more?
Didn't expect such a kind of interactions with the students. We are very happy. And now it's a time for giving the thanks by Dr. Rajeswari, PG coordinator of the department.
Thank you. Thank you.
Good afternoon to all present here. It is on behalf of our Directorate of CPPG. It is my pleasure and privilege to propose formal vote of thanks. At the outset, it is my pleasure to thank our Honorable Vice Chancellor who permitted us to conduct this guest lecture series. And it is my pleasure to thank our Director CPPG for his support and guidance in conducting this guest lecture. And it is my, I like to thank our Dean SPGS PG Studies, who was instrumental in conducting the guest lecture in all the departments of our university after the final Viva OC. And it is my pleasure to thank our Dr. Rahul Kapoor, senior forage breeder, Punjab Agriculture University, Ludhiana, who has given the detailed lecture about the forage crop improvement, forage breeding, as well as the biotechnological interventions in forage crops. Thank you very much, sir. Our students were very much delighted, as well as the understand the forage crop improvement. Thank you, sir. And I like to thank our head of the department, genetics and plant breeding, professors of CPPG, and the student friends. Thank you, one and all.